Oh, great, man. So you uploaded in the Google Classroom?
Yes, so I did that table and then I draw all the amino acids.
Great, great, great. I'll check. So you are familiar with the names, right?
Yeah, with the amino acid names, I can show you.
Can we have a quick go through? Let me ask some questions. Ready for the
Sure.
questions? What does
Okay.
F stands for?
Yeah. Uh, Let me go to my Google Classroom. Phen-
No?
Phenylalanine.
F for phenylalanine. Correct. Good. What does A stands for? A.
Alanine.
Alanine. That's a primary amino acid. Okay. And glycine. Correct. Is the
So
screen
out
with?
of all these amino acids, these are all in the found in our body, right? Like all of these amino acids.
Yeah. See, among them, there's some few are essential, few are non-essential.
Okay, that I did
Okay.
not learn yet.
There is another classification. It's a good question actually. So Adi, you have two sets of amino acids. Okay, might be that question might be your today next homework. But I will explain what is the question.
What are the acids?
Yes, few amino acids are present in our body. They are called non-essential. Non-essential means they are not required in the diet. Clear?
So
Yes.
they are present in the body, but we don't need them for any function.
Not like that. They are not required in the diet. Like we don't want to give any dietary supplement for them.
Oh, okay.
Whereas few amino acids should be given as a diet. Okay, they are called essential amino acids. They are called as what?
Essential. So the amino acids which you don't need to give in the diet is non-essential, but essential is which you need to give.
In the diet. Correct. So you can find the name, like you can classify the name after the class. Like you can take a small homework. Fine.
Okay, so classify the
Yeah. So
amino
quite
acids. Okay.
classify the amino acids based on diet, like essential, non-essential.
Okay. Yeah,
Clear?
I'll check that.
And if you look at the chart, is my chart visible? It's from previous class.
Yeah, the day one notes correct. Yeah.
Yeah, your my notes also with you, right? Like you can able to download the
Yes.
notes. Correct,
Okay.
Adi? Great. So have you gone through all this like classification? Can you see different classification over here? Non-polar, polar.
Positive, negative, and polar.
Correct. Positive, negative. They belong to polar. Good. Okay. What about the yellow color? They are non-polar. Okay. Have you heard of hydrophobic and hydrophilic?
Yeah,
If you
hydrophobic
fear for
means that it is afraid of
yeah,
water. You
if
will
you
say
fear
that.
correct. Fear of water, right? Or I won't like water. Correct. So what
Mm.
happens? Two amino acids are like lipophilic. They love to be in lipids, fat, but they do not belong to water. Clear?
Clear.
Those are called hydrophobic amino acids. So what do you see here, like glycine, alanine, valine, are hydrophobic amino acids. Okay,
So
Adi?
all the non-polar amino acids are hydrophobic.
Hydro no. Yes. All non-polar are called to be hydrophobic. You're right. Polar are called hydrophilic. Clear?
Hydrophilic meaning is
Water loving.
water
See,
loving. Okay.
if I have a love for something, it is called philic. Philic. Correct?
Okay.
Okay. So water loving, water hating.
Okay.
Simple. Okay, Adi?
Yes.
Correct. Okay. So in today's class, we will see even more better than that. Fine,
Yeah.
a bit
So,
technical.
yeah. Sorry.
Yeah.
Also in the last class, you said that you'll like put a book. like some book you'll upload, right? In the
Oh,
drive. I did not see the book.
book is not there. Sorry, I forgot to upload. This time
It's
I'll
okay,
upload.
sir.
Yeah, yeah, yeah.
Yeah.
If anything is missing, no, you can ask me. But I mentioned the book name, I guess, like Leninger. You remember the Leninger book name?
Mm, no.
It's freely available, but anyhow, I'll just upload again. Okay, Adi?
Okay. Mm.
Then what else we have seen in the last class? Make a quick go through, please.
We were also looking about the permutation table.
Okay. You remember about the DNA, like what we have seen about DNA structure,
Ribonucleic acid.
deoxyribonucleic acid. The difference between ribonucleic acid and DNA, correct?
Yeah, also the structure of DNA, which is pyridine and purine.
Correct. Fantastic. So you also can look at the structure, but mainly we focus on protein because high amount of research is focused in protein. Clear? So we'll go with the current trend. Like what is in the current trend, we'll follow that because you are research oriented, correct? You want to
Yes.
go your career, take your career. What is there in the current scenario? That's my aim too. Okay,
Okay.
Adi? Yeah. Fine. Let me go to today's class. Fine. Have we discussed about the primary structure, secondary structure, tertiary structures in the previous class?
I don't think so.
No, we are not discussed.
No.
Okay, we'll discuss that too. And today's class, as per the schedule, it will be about introduction to CAD program. Okay, we'll have, see, actually in the last class we have some few slides left. That slides we'll cover today. Okay, after we cover those slides, we will move on to CAD. Okay,
CAD.
do you
Okay.
know the full form of CAD, Adi? Like I think we have said in the beginning of the class, during our discussion phase before this class were finalized. Like you also in your poster, I have seen what does the word CAD stands for. Any idea? CADD. Okay, I'll give a clue.
Computational drug
Correct.
design. Okay.
Fantastic. Computational aided.
Computational drug design,
Correct.
I think. Right?
Computational aided drug design. Okay.
Oh, aided only. I forgot. Yeah.
So computational aided drug design. Clear?
Correct.
Yeah. So that would be the right statement. So that is very important to know here. Okay, Ari?
Mm.
Yes. Yeah, that we will see. But before that, we'll have some quick go through of the previous thing also.
Okay.
For the delay, my system is loading. Just give me a minute.
Sorry, sir. Can you repeat?
No, like 1 letter code and 3 letter code of amino acids. Have we got a
Uh,
chance to practice that?
Yeah, I think I know it, but like for some, I don't understand. Like, why is tryptophan W? Like that is a little bit weird.
Fine. For that, I'll explain you. What is the 1 letter code for sodium?
Sodium is Na.
Correct, na? Because they have natrium. Getting my point?
Natrium means
Technical term for sodium because we can't give S for sodium. If we give S for sodium, what happens to sulfur then?
it won't work.
Correct. So to create to so avoid confusion, scientists have declared in that way. Clear?
Oh, okay. So there is a
Scientists have,
different meaning for that. Okay.
yes, because same monitor code exists for other stuffs too. That's the reason.
Correct.
Hmm. Okay. So do you have your notes with you? Like however session will be recorded, and I'll share the notes too. Whatever the points I say extra, kindly make a note of it and do a research in it. So you are good in googling, right? You are good in surfing,
Yes,
browsing,
yes,
correct?
mm,
Yeah. So if you are good in browsing, you look for the terms like what I say extra. Okay, Adi?
correct, sir.
Good. Yes. So this is about a CAD for beginners. Like as we discussed, CAD. Before going to actual CAD, we have few slides left from the previous session. Let me finish it fast. So can you see here? Have you ever gone through the word called mutation, Adi? Anytime?
Yeah, mutation in DNA means that it's a variant in the DNA.
Correct. So anything mutates. Correct. If it's
Mm.
mutate, what will happen? The
A
entire
different
property
DNA.
and the entire property may change because whatever happening in DNA will impact in RNA. That RNA will impact in protein. Am I right?
Correct.
So that's why look at the DNA level. So what happened here? So before that, have you ever heard of the term called DNA code, triplet code?
Um. Yeah, like that. The base pair, right? Like how it's the bond
Yeah,
between
I
them.
think
It's
the
a
last class. Do you remember the last class we had discussed that? Yes, Adi.
yeah. Okay.
Triplet code. Like I also shown you. That's the reason we need to go through the basics. Like AAA make code for a particular amino acid. Correct? Yes. Now we are going to look at the mRNA level. What happened? There was a small change. See,
Okay.
instead of
Sir, you're in mute. Actually, sorry, I can't hear you. You're still in mute. I'll go out and come back in.
How about now?
Yes, sir. Now you're audible.
Yeah, thank you. Thank you so much. Yes. So I'm saying about the mutation, Adi. Okay, Adi.
Yeah, okay.
So there are three types of mutation. Okay. For
Are
example,
you
if you,
sharing your screen?
yeah, I'm sharing. I'm sharing. Hope it will be loading. Just give me a second.
Yeah.
It should be loaded now. There are
Yes.
three types of mutations. Okay, have it in mind. Something is called silent mutation. Another thing is called nonsense mutation, missense. The reason is, okay, you can make a note of. You will get the notes. We just make a note of the reason. When do we call someone as nonsense? This is a way we scold people, right? Don't speak
Yeah,
like a
like stupid like that. Yeah.
yeah, stupidity. Not at all relevant to the particular sense. Correct. When you
Yes.
guys are talking about cooking, suddenly one guy is saying about some other about some drainage something. Correct. It's irrelevant to the point. Same way,
Yes.
what happens? See, uh, when you go to the triple letter code, okay, instead of a triple A, if it is at least AGG, what happens like TTT, triple T, if something is replaced with one single letter, look at here, it is TGC. Okay, nothing
Okay.
wrong. Yeah, yeah, nothing wrong, right? Like TCC and TGC, only one letter how code have changed because instead of one amino acid, another amino acids have formed. Correct, arginine, threonine have been formed. But
Okay.
what happened here?
No amino acid is formed.
No amino acid is formed, so it become truncated. It become what?
Truncated.
Truncated. See, at least in this case, the activity of the protein might have reduced. The efficiency might have reduced. So it is acceptable. But in the nonsense, what happens? It in the completely the protein will get truncated. Clear?
Okay. I have one question, like in the previous
Please, please.
slide.
Yeah, yeah, yeah.
So this is silent mutation.
Hmm.
The DNA and the mRNA level, like I know just one letter is changing, but how come
Hmm.
the protein level is still
Yes,
the,
because we have only 20 amino acids. Okay, sorry, we have only 4 letters, Adi, right? But they
yes.
have to form 20 different amino acids. So there will be some overlap. If you want to recall the previous slide, let me show the previous slide also. Do you remember our previous slide? Do you
Yeah,
have
yeah,
your previous slide? Can you please go to the last before slide quickly? Can you open it in your computer and see?
yeah,
Like
that the table, right? The pink color table, that one.
the pink color table, the table which says about yeah, second letter, correct?
Yes.
That is called codon table, Adi. That is called
Codon
what?
table. Codon table.
Codon. Yeah, codon table, where I have told about permutation combinations, like
Yes.
how the word Adi can be arranged. You remember that?
Yeah, I wrote.
So nearly, if you go back, look at arginine. Have you opened the slide?
Yes, I'm having it in my laptop.
Look at arginine, ARG. So how many codons are coding for that?
4.
4 codons are coding for that, correct?
Yes.
Because different combinations may lead for one amino acid. But what if entirely different amino acid has stopped forming, like UGA instead of UGU? If UGC is formed, look at the right corner, right? UGU,
Yeah.
UGC, UGA. Okay.
So they're all truncated.
In the means UGU instead of UGU, if you are having UGC, look at the right corner, top corner. Can you see that?
Yes.
UGU, UGC in your slide. Can you see? Like the
I
pink
have.
table. The pink table. Slide number 17 in our previous notes. Day one notes.
Yes, here only I'm sharing.
Uh-huh. You are presenting, Adi?
Yes, I have it open.
Yes, okay, okay, fine. Yes. Yes, exactly, exactly. Can you see that? The top corner, UGU, UGC, correct?
Yes.
So that it is called silent mutation. Am I right? Why it
Yes,
is
sir.
called silent
Correct.
mutation?
Because just one letter is changing, and there is only so many combinations that you can make. So that's why it's
But
coming.
even one letter have changed, there is no change in the amino acid. So it's silent. The mutation is not valid. But look at the second bottom, like instead of UGU, what happens if first U is replaced with CGU, where I've
Um,
underlined near
C,
arginine.
CG, yeah. That is silent.
No, UGU to UGC is silent because both are coding same amino acid. UGU to CGU, that is what?
Stop.
Missense.
Oh,
Missense.
yes. Oh, okay.
Getting my point? Not
Missense. Okay.
missense. Okay. Whereas completely it is, if it is UGA, that becomes stop codon. Clear? That becomes nonsense. Now come back to my slide. Got my point?
Yeah, what is conservative and non-conservative?
Yeah. So you might have heard this term like conserved family, like our system is a conserved one. Means they are retaining for a lifelong. They're retaining for a generation
Lifelong.
after generation.
Yeah.
See, we being Hindus, I do not know. You pray God, right? Like
Yeah.
Hindu God.
Yeah.
So even you are away from your homeland, you're still praying, right?
Yes.
And you might have seen some families, they settled in US for many generations, like 3 to 4 generations, but still they follow their native routine. Correct?
Yes, correct.
They are called as conserved family. But in the
Oh,
case, yeah, in the case that is generalist term. In the case of molecular biology, the word conserve stands for retainment of a particular codons or nucleotides. Okay, here these amino acids. Can you see? Can you guess the amino acid? What is N?
N.
So in the next class, I expect you to be even more strong. Okay.
Okay. Okay, let me just find it quickly.
Can you see my slide? What I have written?
Yeah, I can see. You just
Aspargine.
put each one is N.
Yeah, aspargine. Can you see?
Aspargine.
Yeah. Okay. So this
Yeah.
aspargine is conserved. This aspargine is conserved. Imagine these four sequences are four different organisms. Four
Okay.
different organisms.
Organism. Yes.
Uh, for example, the hemoglobin protein in blood will be different in different organisms in some points. Let us humans, monkey, chimpanzee, okay, orangutan.
Mm.
Clear? I'm giving
That's
some
it.
example. And what happens? See, so this asparagine is conserved. Whereas what about phenylalanine, tyrosine? This Y is called
It is
tyrosine. Okay? F is for
tyrosine.
Y is tyrosine, F is phenyl
Phenylalanine.
phenylalanine. Okay. So here it become non-conserved, unconserved, non-conserved. Clear?
Clear.
Yeah. Okay. Okay. What you want to understand is a different species will have different sequence. That was speciation occurs. Got
Okay,
it?
so each one will vary. Yeah.
Yeah, it is varying. What happened? Suddenly a lysine got introduced. So K for
Suddenly,
an amino acid got introduced over the time period.
oh, okay.
The reason is, imagine your grandfather and his great grandfather was staying in the land. Your grandfather decided to climb the hills, like he's staying some 2000 feet above the land. Okay, seashore.
Oh,
He decided to imagine he decided to stay over there, and your father was born there, and you was born there, and you have five generations. You are staying in the hill.
mm.
What will happen to your hemoglobin level?
It will be different than the people who was living in the floor
Yeah, why?
because of elevation.
Because of elevation, there will be less oxygen.
Yeah. So less oxygen means less hemoglobin.
No more hemoglobin should be there. Why?
Oh, because
Think.
hemoglobin is just carry oxygen, right?
See, being in plane, we are having more oxygen content.
Mm.
It is abundant. It is an abundance, correct?
Yeah.
So you can take any time. So less hemoglobin is enough. But very simple. See, my hands are free. I can grab things anytime, correct?
Yeah.
What if your shops are going to close for next 10 days? What you will do? You take more stocks, right?
Yeah.
Same way. So if you are staying in the uphill, your body will evolve, your grandfather will evolve in such a way to have more hemoglobin so that you will capture even the little oxygen available.
Oh, okay. Yes.
Can you able to think that, Adi?
Yeah, because what you're trying to say is a higher elevation, there will
Ah,
be less oxygen, so there will be
ah.
more hemoglobin to capture that less oxygen.
Exactly, exactly. That what species will start evolving themselves. Okay.
Okay.
See, if you want to learn biology, biology is such an interesting subject. If you learn basic biology only, you can understand molecular biology and you can think in the perspective of research. Fine, Adi?
Yes.
So
Yes.
you want to think, you want to watch. See, whenever you want to excel in research, you watch a lot of Animal Planet,
I
National
watched
Geographic.
a video for transcription.
Mm-hmm.
Like it's a Khan Academy thing I saw.
Khan Academy, you can see. You can, like Amoeba Sisters. There are a few people called Amoeba Sisters. You know that?
It's
Amoeba,
Amoebas.
yeah, Amoeba Sisters. They will also make good animations.
Wait, I
And
will
I'll
check.
share few links after the class. Now I'll share some few good links of biology.
Like
Okay,
this one.
exactly. Amoeba Sisters. The yeah, you like anime? It's not a great anime, but you will learn them. They will explain a lot of in Cartoon Network way.
Use of numbers, protein synthesis. Yeah, I'll understand from this.
Definitely, you can understand because it's like a story.
Mm. Okay,
Fine, it's like
next.
a story, and you can catch all the points from here. Like what is chromosome, why is chromosome, what is gene. These all because your level, your grade is less, but your aim is high. Correct.
Mm.
So you want to catch up with all these people. Clear,
Okay.
Adi? So you
Thanks for telling me this channel. I will see in my free time.
sure, Adi, sure, Adi. You go through all this. Okay.
Oh, okay.
Yeah. Get back to the work. So you got it. Why speciation should happen?
Okay. Yeah. So
Like that
typical.
each and yeah, each and molecular level we have to understand, right? So our alphabets are so hereafter when someone asks what is A for not apple, A for alanine.
Alanine.
So you have to say in that way. You have to get practice with all these terms.
Yes,
Clear?
I'll practice. Just one
Great.
more thing. Just like
Yeah.
how it's being ordered, it can be taken away also, right?
Yes, exactly. Look at here. You are right. That's the next step. What happened here? Can you guess what is L? L is leucine. Okay. The leucine got removed. Leucine got what? Removed.
So is this an essential amino acid or non-essential? Like,
That is not a concern here. Okay.
oh,
Here, okay, here, whenever the protein is there, the protein will differ. So I'll say some one of the important mantra. The important mantra is sequence to structure, structure to function. Can you see here what
S
I've done?
a
Sequence to
sequence
structure,
means a sequence
sequence.
is structure. Okay.
Okay, sequence to structure, structure to function.
Function.
Very simple. See, I have a brick. What a
Hmm.
brick is made of?
Brick is made of concrete.
No, brick is made up of some mud, sand.
Oh, yeah, actually they use concrete bricks, but okay.
Fine. Oh, concrete. Fine. So again, there is some kind of cement. Am I right?
Oh, okay.
So cement is some granules. Can I call it granules?
Yeah, okay.
So with these bricks, I can form any structure. Am I right?
Yeah, I can put
I can form
anything.
a statue too, right?
Yeah,
I can form a table too. Correct?
anything.
Yeah, I can form even a wall.
Hmm.
If I arrange in this way, it will form a wall. So my question is, this is a sequence and forming different structures, and the structures can perform different functions.
Okay, yeah.
Getting my point?
So that sequence will form a structure, and each of that structure can form varying functions.
Varying functions. See, look at different types of protein. These are all proteins.
Yes.
Whatever you see are sources of proteins. Like fish. Okay, fish protein, muscle protein. Generally, this protein term is known for food, but they like general public won't understand the protein is something which is running our body. It is like a fuel for our body. I'll put a very right word. Proteins are building blocks of our body, correct?
Building blocks. Yeah, that makes sense. Correct.
Proteins are building blocks of our body. And another important term is proteins are also called as workhorse of our body.
Workhorse.
Why? Oh, this workhorse is a very common term because in kingdoms they use only horse, right?
Mm-mm.
Imagine a kingdom without horse.
It won't go good.
Go good, right? They have to do a lot of functions like transport. So horse is important. Same way, very simple. If I have a candy, if I chew a candy, or if I have right, salivate my candy, what happens? Immediately an enzyme called salivary amylase secretes and try
That
to
is
break.
how this saliva will come. That is how.
Exactly, saliva will come, and saliva consists of a protein enzyme called salivary amylase. Okay, so that
Salivary.
enzyme is salivary amylase. Okay, that
Mm.
enzyme will start breaking the polysaccharides to disaccharides. Our
Okay.
glucose will start broken when you chew something, or some wheat, or some, if you are eating some rotis or something. Got it, Adi? So it is very, very important. So enzyme is also a protein, and your hemoglobin is a protein. Your hair is called keratin, that is a protein. Your skin, collagen, that is a protein. Don't worry about these names. But what in simple is
Mm.
everything is a protein.
Yeah,
So
everything.
without your proteins in your body, you can't function properly. Clear?
Clear.
So each and every protein is having different function.
Mm.
Okay.
Okay. Mm.
What's the function of a blood?
Can you repeat, sir?
What is the function of the blood? Blood.
Blood is used to transport oxygen through the body.
Fantastic. So blood is having different function. They have to transport oxygen, correct?
Yes.
What is the function of muscle?
Mm.
Without muscles, you can't do any you can't do any movements, right? You move your hands because of muscles. You lift something because of muscles,
The
correct?
muscles we will say it's used for motion.
Yeah, muscles inside your body is using for motion,
Okay.
correct?
Yes. Mm.
Locomotion, movement, everything, muscles are important. So this is also made up of protein. Like all it is made up of protein inside.
Mm.
So if I'm fat, my fat will be outside the protein, but protein is the core.
But the fat in our body is also considered to be protein, right?
No, fat is something different.
Oh, okay.
See, again, that's what we need as some basics. What do I say? Just a minute. So I'll make it faster. If we are running out of time, I'll make it fast. See, very important. You asked a very important question, like whether protein is also fat or not. See, 4 important molecules are there in your body. They are called macromolecules. Okay. How
Okay.
many molecules are there, Adi?
4, and then we call it as a macromolecule.
One is DNA or RNA. They are called
Mm.
nucleic acids.
Yes. Okay.
DNA or RNA.
Mm.
Nucleic acids. Okay. Nucleic
No, so
acids.
nucleic acids is number 1. Okay.
Yeah. They are very, very important. Nucleic acids are the number one thing. Because nucleic acids only carrying the information for how organism should look like. They are like a blueprint.
Okay.
The second important thing is carbohydrate. In common terms, we call them as sugars. You remember carbohydrates are in lot of now you can see some dietary what advertisements they call carbs.
Stop eating lots of carbohydrate. Like,
Correct. Carbohydrates. So carbs, they are also part of the body.
okay.
Okay, they also make a very important function in our body. So second important macromolecule is carbohydrate. Clear?
Correct.
The third important is our protein. Okay, which are made up of amino acids, which are made up of
Amino acids.
the sugars and another than glucose. You can browse through this. You can try all these terms.
Okay, oh, you want me to write? Okay, I'll write.
Okay. Yeah. The fourth important thing is called lipids. Lipids are technically technical terms of fatty acids.
Oh,
Clear?
yeah.
So this is different. This is not a protein. Lipid is different from protein. Clear?
Okay.
See, if you see some soft tissues, like for example, in our buttocks, okay, or
And
in
that
our
must.
cheek, they have fat. Even if I have a belly, that abdomen is a fat.
Okay.
Do you like you're vegetarian or non-vegetarian?
Vegetarian.
Vegetarian, okay. But anytime you have seen some slaughterhouses, you got a chance to see some slaughterhouses,
Yeah.
or you can see even on the internet, they will see some white color wala. When the mutton is slaughtered, go to slaughtered or slaughter, okay. This is science. Don't think it bad. You can see some
Oh,
white color tissues. Fine. Those white color tissues and another than they are fat.
oh yeah, I can see now.
Correct.
Hmm.
So that is fat. Whereas the red tissue or the brown tissue, that is called protein. Yes, Ari.
Yeah, that's what the people eat.
That's people eat. People also eat fat, but fat is not good for health. Not overeating fat is not good for health. Healthy fats are there. Okay.
Yeah, like in the nuts and everything.
Nuts are have healthy fats, and even you can eat carbs in a regularized way, not that fiber carbs, not a very loose carbs, simple carbs like glucose. You should not eat more candy. If you're eating having more candy, what happens? You are having a simple carb. Okay, that is not good
Because
for health.
sugar is bad.
Yeah. So we will come back to this, but
What
these are
is
the
macromolecule exactly? Like, if you have to define it, like, what will it be?
see. Actually, I want to go in a systematic way. You know atoms.
Yes.
Can you say define? Can you say some example of an atom?
Basic building block of all matter.
Yes. If I say sodium is an atom, correct?
Correct.
This
Gold
atom forms molecules. This atom form what?
molecule.
Example, carbon is an atom, whereas methane
See,
is a molecule.
yeah.
Methane is what? Why it is a
Like,
molecule? Because it is made up of carbon, hydrogen, correct? Carbon,
yeah.
one carbon, four hydrogen, correct?
Yes. Okay.
Whereas glucose is a macromolecule. Do you know why?
Because
C6,
it's not simple. That's why.
it's not simple, but it's even a huge number. Like they're the part of the body. Like, and it is very complex to as you assume. This is glucose structure. And one it is not one glucose. Actually, I draw glucose like pyranos. Okay. Many glucoses assemble themselves and they
Okay.
form polysaccharides. See, these are very huge terms, Ari, which people study in colleges. Okay. But to your level, I can try to make you a bit of
Understand.
understanding.
Yeah, I'm understanding.
Sandosham. You understood, right?
Yeah.
You should start in that way. Okay. Slowly you start. You have a lot of time, but you spend all your energy to know about the basics. Basics are
Yeah.
very, very fundamentals are very, very important. Okay, Ari?
And nowadays I'm watching Khan Academy only, sir. That video.
What, Ari?
I'm
Nepal?
watching Khan Academy translation video only.
Yeah, you watch that, you try to understand what are the parts of the body, what is an organ, what is tissue, what is cell. Okay, slowly you try to complete all Amoeba Sisters videos. Okay, and also I'll say send some amazing animation videos. You try to understand that. Okay, when time
Yes.
permits, but follow one video. You write the concepts. Okay, and biology is very, very interesting field. It's learning about ourselves.
Yes. Okay, sir.
Yeah. So without further delay, move forward. Right. See, among the four, which is very, very important for us?
Proteins.
Yeah, why it is important?
Because
Because
it's a running horse.
Yeah, it's a working hours. Very good. You just
Hmm.
got the word. When if anything it goes wrong in this working hours, whole body will form disease.
Cancer. Oh, okay.
Yeah, lot of disease, not only cancer. Look at here. See, just one mutation, Adi. Can you guess the number in mutation? Here, I made a circle. Can you see here?
Mm.
Okay. In next class, you have to be thorough with the name. Just now I told N stands for
Um,
just a couple of minutes ago. Asparagine. You remember?
aspargine. Yeah.
Y stands for tyrosine. Y stands for
Tyrosine.
yeah. What happens in the 501 position, like 501 position? This asparagine have transformed into tyrosine.
Oh, okay.
Asparagine have transformed into what?
Tyrosine.
So it causes a disease.
Mm.
Okay.
Okay.
There is another interesting medium thing called sickle cell anemia. You okay? Take on homework. This is your homework. Mechanism of action of sickle cell anemia.
Sickle
Cell
cell
anemia.
anemia. So you just want me to look at that in Google, right? Okay.
Yeah, you want to look at it in Google and you want to make notes of it. What mutation occurred in sickle cell anemia?
Between which two? Okay.
You can make it a question. Impact of mutations in sickle cell anemia. That's your question. Please make a note of it.
Uh,
Impact of mutation.
in sickle cell anemia.
Fantastic. That's your second homework. What's your first homework?
Um, I'm supposed to classify all the amino acids.
Essential and non-essential. Okay. Essential and
Non-essential.
I'll put in your Google Notes also. Okay. Sorry, your WhatsApp chat also. You can type the chat. You can type the questions in the chat itself directly. You have your web WhatsApp open.
Actually, Amma is having a phone. She went to Google
You can
Notes.
write in your book and say Amma to post that. Okay.
Oh, okay.
To have a record.
Mm.
First homework is essential, non-essential amino acids. Second homework is about.
Impact of mutation in sickle cell anemia.
Then you will try to understand that. Okay. Look at other interesting examples. Just again, one small defect in the protein will lead to a disease called Alzheimer's disease. This disease is so famous in US. Have you heard of this disease?
They will forget, then they will not be able to move like this.
Correct. Old people will forget, like adult people will forget what is happening. They will have memory issues.
Mm.
Okay. Again, there is another protein. It got phosphorylated. It becomes cardiac injury. Heart will stop functioning.
Oh.
There is another protein. If get affected due to infections, it will become meningitis. Your brain protective layer will cleave it off. Your brain will stop functioning. Clear?
Yeah.
And there is another protein called cancer, KRAS. What happens if the protein got malfunction? It will become carcinogenic. A tissues started proliferating. You know about cancer, Adi?
Yeah, cancer means that the cell, it will keep on splitting until there's too
Correct.
much. So,
Proliferative growth. Okay, it's called proliferation. In Tamil, it is called புற்றுநோய்.
புற்றுநோய்.
Yeah, புற்றுநோய் means what? You know, எறும்பு புத்து தெரியுமா உங்களுக்கு?
On that hill, like ant hill, we'll say, right?
Ant hill, fantastic. And the ant hill எப்படின்னா, like ant will keep on accumulating. It won't stop. It will keep. If you leave it, no, entire tree will be ant hill. That's why our ancestors have named cancer as புற்றுநோய். If you leave, if you leave the cancer to grow, it will start accumulating all over your body. But people won't survive for that. In the midway itself, people will die.
Yeah.
It's a very challenging disease. If you work on cancer, no, again it is very being in US, no, you can focus either on cancer or we can focus on Alzheimer.
I'm more interested in Alzheimer. Like.
Great, man. Then we can we plan a project in that. Okay, Adi. And then it will be a great groundbreaking. I have more research articles. Just I publish myself in my about Alzheimer. You can go through my channel. Rather saying channel, it is called Google Scholar. I'll put in the WhatsApp group.
Yeah, I know Google Scholar. That's very nice, sir. Congrats.
Thank you, man. Thank you so much. You
Uh,
found my Google Scholar?
yeah, what is your Google Scholar name?
It's Chandrasekhar Ponnuswamy. Like I'm putting in chat.
Okay.
You just click my chat.
Sir, so many papers.
Again, I'm a budding researcher. I have to achieve a lot, but to my level, it's fine. Okay. Yes. And I wish you also get a paper soon.
Okay, sir. For that only you should help me.
Yes. But you have to work hard and you enjoy the progress. Okay. You
Yes,
enjoy the
sir.
progress.
This is fun. I like it.
Yes. You're fine. Now, so far, last two classes, we are talking about proteins. What is protein? What it is made up of? Am I right, Adi?
Correct.
What scientists will do while trying to understand the protein? Protein is something we can't see by eyes. See, you can see muscle. Definitely, if you see some muscle, that is a collection of the entire system. But protein is something a macro molecule, which you can see
Oh,
only you can't see by eyes.
okay. Yeah.
You can't see by eyes.
Nice.
Very simple. Uh, I think I told this example. You cut this dart into 1,000 pieces. I told this example, right?
Yeah, that grain of sand you said.
Yeah. 1 millimeter into 1,000. Then that becomes 1 micrometer. Correct. 1 micrometer by 1,000. That becomes 1
Nanometer.
nanometer. Correct. Generally, proteins are in nanometer scale. Proteins are in
Nanometer scale.
nanometer scale. So we can't see by our naked eyes. I'm
Only
saying
through
about,
scale, right?
yeah,
Yeah.
I'm saying about one small molecule. Again, that molecule will accumulate themselves to become a big molecule. Correct.
Hmm.
So what scientists will do? Scientists will try to make a crystal out of it.
Crystal.
Okay. Crystal, like a salt
Which
crystal. Can you see?
no, which we can see or not see with the naked eye.
We can't see, but we know that's a pure form of protein. Nothing is there is no impurities.
Hmm.
Scientists will
Okay.
try to make crystal. Okay. Then what scientists will do? They will just bombard the electrons, or they will bombard the X-ray over it. They'll bombard the X-ray over it, and they will get a lot of diffraction pattern. Have you seen any time any X-ray images?
Yeah.
Have you seen X-ray images, right? Like how X-rays pass through the body and just show which is opaque, which is right. Same
Yeah.
way, a kind of that, but it's not exactly like that. But it's like X-ray crystallography. I'll share the slides. You can go through the slides. Okay, one by one.
Great.
Then they will try to understand each and every atom in it. They try to understand
Each and every atom in it.
each and every atom in it. So today is in end of the class. Like we are nearing the class end, but I'll show some 5 minutes. We will
Ah,
demonstrate it. Okay, about how a protein will look. Then
okay.
this electron density map they will solve with some softwares, and they will try to understand the structure of the protein. They will try to understand the
Structure of the protein.
protein. Okay, Ari. Yes. Yeah. First, I will say this, and then I'll go back to this guy. Yeah. Now you can see here, Adi.
And that um,
Yeah. This is what.
hydrophobic charged polar glycine
Yeah.
cross linkage.
These are the types of amino acids, and we are thorough with this. This is called primary, Adi. This is called as what?
Primary.
Primary structures. Okay.
Hmm.
This primary becomes they will assemble themselves to form tertiary.
That
Okay,
primary is secondary.
yeah, primary assemble them and they will form a shape that is called as what?
A helices and B sheets.
Alpha. Yeah, A means alpha helices,
Oh, alpha, beta. Okay. Yeah.
beta sheets, alpha helices and
Beta sheets.
Then they will form a group, and they become tertiary structure.
Oh, okay.
And several tertiary structures will become quaternary structure. So tertiary means one chain, and quaternary means multiple chains.
Okay.
Clear? One chain, and quaternary will be what?
Quaternary means
Multiple chains.
so
One
4
chain,
and above we will say, right?
not 4 and above. Each and one and above itself a quaternary.
Or
See,
like in max we will say, right? Like monomial, polynomial, that one.
yeah. I'm not going to that level, but to biological level, what I know is one chain is monomer. More than one chain, it becomes quaternary. Don't think about quadrants and don't confuse yourself.
Okay.
Primary is just linear compounds. Okay, primary is just linear. Okay, like a rope. I try to make different shapes out of the rope. What are the two shapes I made? Look at here. What are the shapes I made? Like alpha helix. Can you see the shapes? I can
Yeah,
make a
one
shape.
is straight and then one is
Yeah,
curved.
rather saying shade is one is like helix. This is called alpha helix. Is my screen visible, Adi?
Yes, I can see.
Another thing. Can you see the shape? This is the helical shape.
Yeah,
Yeah,
curved.
another is like a pleated sheet. See, if you take a paper and you fold like that, you have a fold. If you take a paper now, like, see, I'm taking my sheet. Okay, I'm folding like this. Then I fold again like this. Can you see the way I
Yes,
folded?
like you're holding a mouse pad, yeah.
I'm folding my mouse pad. See, again I folded like this. Again I folded. This is called pleated folding.
Pleated. So one time you're folding,
Hey,
the next time
yeah,
you're pleated.
your mom might have wearing saris.
Ah,
She will have lot of pleats. Correct?
yes,
She
like
will
that
fold
type.
the sari. Right? She will fold the sari. That's called pleats.
Yes, okay.
You got it now?
Yes.
That is called pleats. Getting point. Pleat is like folding like this. So it may be a pleated sheets, beta pleated sheets. Beta pleated
Beta
sheets.
plated sheets.
Yeah. Sorry for my writing. Okay. Beta pleated sheets. Again, technically speaking, alpha helixes will be having intramolecular hydrogen bond. What they will have?
Intramolecular hydrogen bond.
What is called intramolecular?
Within the
If
molecule, there is a
within
hydrogen
the
bond.
molecule, fantastic. Within the molecule, the hydrogen bond will be formed. Clear?
Oh, correct.
What about beta sheet?
Beta sheet is just like a type of folding that is done. The
Yeah,
pleated
but it is intermolecular. It is what?
within the molecule.
Between the molecule, not within the molecule.
Okay, between, okay.
This is between, this is within.
Between and within. Great.
Yeah, within is alpha, between is beta.
Within is alpha, between is outside of the molecule, will we say? Or?
Yeah, different. The inter means like different. For example, international is between different countries, right?
Okay, yes.
It's Indo-China, Indo-US. That is international. It is within Kerala, Tamil Nadu. That is intra.
Intra, so
Yeah, you
between.
got it. You got it. Look at the hydrogen bonds. That's what I'm trying to say.
I'm seeing.
Can you see the circle what I made?
Yes, you're making that between the purple color picture is in the top and blue is down.
Correct. Got it, Adi. Okay, now let me introduce this gentleman. Okay, he's a Malayali, Kerala, and he's a very pioneer in our whole world. Okay, he is the person, Dr. G.N. Ramachandran. Okay, the scientist, what he discovered is he found he made a chart, he made a quadrant. And he tried to understand where the protein will lie. Where the protein will
Lie.
See, we are a scientist. We are trying to understand the protein by doing a model, correct?
Okay. Yeah.
You made a protein docking, if I'm not wrong. In your poster,
Yeah.
you made a
It's stuck. It's stuck.
H dock, right? Before you dock the protein, if it is not available, if it is not done by experimental biologist, you want to do a model. You want to do a
Model.
that model you have to validate. The model you have to
Validate.
where do you validate? You will validate only through Ramachandran plot.
Okay.
Validate through what? Ramachandran plot.
This plot is very confusing.
Don't worry. I'll make it very, very simple. Let me make it very simple. This plot is otherwise called as phi psi plot. This plot is otherwise called as what?
File side plot.
This symbol is called phi RE. This symbol is called phi.
File.
And this symbol, no, like a trishul. Can you see a trishul here?
Yeah. Yeah.
That is psi.
So file
To make
side.
phi psi plot, it is otherwise called as what?
File side.
Phi psi plot. Clear?
Okay.
Don't panic about these terms. I'll make it very, very simple. Just you have to just see, if you can't understand in one instance, you just get the recording, listen again and again. Okay.
Okay.
Man, I want to appreciate, my hats off to you because you are studying a college science.
Okay. Yeah. I think I understand.
I understand. I just appreciate you. You're trying to
Oh,
understand college science. It's more than your level, but you try to grasp the things. Okay.
yeah. Yeah.
I can't lower my level too. If I lower the level, then I won't justify your intention.
Yeah. I understand.
Uh, just look at here. Let us make it interesting.
Dr.
Who is
Chandra,
this?
sorry to interrupt your discussion. It's a
Ah,
gentle reminder. It's 8:30.
yeah, sure. So we can take till 8:45, right? Ms.
Yes, we
Bhavani,
can. Yeah.
yeah. Thank you so much. Yes. So Adi, come back. Okay. See, these are the amino acids, Adi. These are what?
Amino acids.
If I want to expand this amino acid, I want to do, let's split this word. Amino acid, okay? So it consists of, for example, I'm trying to write alanine. I'm trying to write what?
Alanine.
I asked you to draw the structures. You remember? No?
Like the structure which I drew, or
You drew.
but I'll just show you.
No, fine. You draw carbon, hydrogen, oxygen. You're drawn. Exactly, man. Superb. Now try. I'm trying to. I do not know whether you understand the each and every components of it. If not, listen properly. I'll make you understand that. They will have a generalized carbon called alpha carbon. What they will have a center carbon called
Alpha carbon.
that alpha carbon will have a carboxy terminal. Okay.
Yes. Okay.
And they will have an amino terminal. Clear?
Okay.
It can be either NH2 or NH3 plus. Okay.
Yeah.
COH. Yeah. Amine and acid. That's why it's called amino acid. Clear?
Amino acid. Yeah.
One amine is there, one acid group is there. One
So
amine
yeah.
is there. So that's why they got the name called amino acid. Clear?
Correct.
And there is also another H group. There is also another H group. Hydrogen group is there.
Yeah.
And the fourth group, because what is the valency of carbon?
How many valence electrons are in carbon?
What is the atomic number of carbon?
6.
Very good. So what should be this valency?
The number of valence electrons in carbon.
How much?
4.
Fantastic. Why? How it is 4?
So because in first cell 2 is there, and in second cell can only accommodate 8. So 6 minus
Good.
2 is 4.
Fantastic. You got an answer. So they have 4, correct? So carbon at the mass can form 4 bonds. Am I right?
Correct, because that's how many valence electrons it has.
So the fourth valency should be an R group. The fourth valency should be what?
R group.
R group. Clear?
Okay.
Right. Now, this R group differs by each and every amino acid. For example, here it is with just CH3.
CH3.
Yes, this R group is, look at your alanine structure. What you have drawn in your notebook.
It's looking like H, like this. It's looking, because which one is it?
No, alanine. Where it is alanine? Yeah, you are right. See,
There is a
you don't.
O below.
No. That is you are expanding that COH.
Oh.
See, instead of the COH, what I can write is C double bond O minus. Why?
Oh, yeah. Because there is two one. There is one more O ion. That's why you're keeping.
That become an ion. If the
Yeah.
H is lost, then I'm putting in ionic form.
Yeah,
Got my point?
because the negative charge is what is coming, right? We take that.
You have written the ionic form. You have written the Zwitter ionic form of the amino acid.
Yeah,
But what I have wrote is a neutralized form.
neutralized form.
You look at again. Look at the amino acid with this neutralized form. First, you learn that. Then only you go for ions.
Yes.
Okay, Adhi, you got the point first of all.
Yeah, you're just expanding upon it.
What you have to understand is coming back to here. Now you got COH. So which is the C alpha carbon? See, this class may look tough, but it is very easy class. By understanding this only, you can understand the entire practicals.
No, I understand.
Look at the circle I made in the center.
That is carbon.
Which carbon it is?
Alpha.
Alpha carbon. What you should say?
Alpha carbon.
Okay, what is the alpha carbon of that alpha man who is standing like Aquaman? You know him?
No.
You do not know him. My God, huh? You have not watched any Hollywood movies. You won't watch superheroes movies, huh?
No, sir. I only Tamil movie watch. That's it.
Tamil movie, fine. Okay, Tamil movie, great man. I appreciate you again. In Tamil movie, you watch a lot of God movie, like they carry Trishul, right?
Trishul.
You know Trishulam? Like if you went to Amman temples and Lord Shiva, what he's having?
This is
This is called Trishul, okay?
like that man is having, right? Like that he's having.
Yeah, in technically it's called trident. This is called as what?
Trident.
Okay, he's a Greek god, man. He's an in Greek, Greek people, Greece.
Europe.
In Europe, they are very ancient people. Okay. See, today, whatever biological term we have, they are from Greece and Latin.
Okay.
And few terms are in Sanskrit and Tamil.
Mm.
Do you know what is the scientific name of rice?
No.
Oryza sativa. Rice scientific name is Oryza sativa. Okay. அரிசின்னு சொல்லுவாங்கல்ல.
அரிசி. That is where it's deriving from.
அரிசி தான் ஒரைசான்னு வருது. Like in Japan, it become O. Like ginger, gingiberacea. It is called gingiberacea. It is inji transform into gingi.
Oh, yeah, that's all nice. Yeah.
I'm not making any my own terms. It's in science facts.
No, I'm checking now. What you're saying is correct. Oryza sativa only is there.
Okay, like same way, lot of Latin terms were there. So this Greek god is called Poseidon. You can check Poseidon.
Okay,
Greek god Poseidon.
Greek
god Poseidon symbol.
Poseidon.
P-O-S-E. You're getting the point?
Yes, it's like he's octopus in the ocean.
He's having a trident symbol, correct?
Yes, correct.
Why I'm saying all this stuff is never forget this psi have derived from Poseidon.
Okay, yeah.
This psi is used in our place for this reason. Actually, if you go for higher classes, you will learn something called water potential. Water potential will stands for solute potential plus solvent potential. Okay. pressure potential. I don't want to confuse this, but
Okay.
simple, psi is something between C and C alpha and CO minus. Clear?
Okay.
C alpha and CO minus.
Yes.
Yeah,
Okay.
clear? That is psi. Clear?
Okay. Yeah.
What about phi?
Phi means that it will be that W. It will like no, not W. That
Yeah,
negation.
CO minus and NH3.
Yeah, it look like the symbol which you're writing next to. Yeah, that one.
Phi. Phi. Clear?
Yes, correct.
Now look at here, Adi. Look at here. This is same sci-fi. They are like a what do we call, like a chart, like this. See, N hydrogen, it will move like this. You're getting my point?
Yes. Okay.
See this rectangle. Imagine they are like a plane. They are like a
Plane.
this plane will move all together.
Okay. Okay.
They can't bend. They can't what? They can't bend.
And
They can't what? They can't bend. They have to move all together like this. They have to
that's all.
move.
Okay.
So this gentleman Ramachandran, G.N. Ramachandran have found. Now you can understand this. They are trying to understand the diagonal angles. How phi psi are allowed.
So using that a plot he's taking, like which one is longer?
He mathematically made a plot.
Oh,
What are the possible ways the phi psi can permit the degrees of freedom? Okay, the phi psi can allow the molecules to move.
okay. Yeah.
You can find this. What is called Ramachandran plot? What is phi psi plot?
A 2D map used in biochemistry to visualize energetically allowed in the backbone angles of amino acids.
You also will understand an important technical term called stereochemistry. You can check this. Stereo hindrance or steric hindrance.
Branch of chemistry that studies the three-dimensional spatial arrangement of atoms.
Yeah. So it'll understand how much a molecule can move itself, how much space is available for the molecule to move. See, what if you're sitting in a corner of the room? Can you able to move your hands freely?
Yeah.
You're in the center, man. Go to the wall.
Oh, then
Go
I
to
can't.
the
That is blocking.
that is called hindrance, correct?
Yes.
Like a lizard, if you are catching the wall, you can't move your hands forward.
Yeah, because so you're on the wall. Volume.
You are on the wall, correct? You have steric hindrance. Now you have a lot of degrees of freedom. You can rotate your hands like this, correct?
Yeah.
But what if I'm near the wall? I can't move my hand. That is called hindrance. Correct?
Yes. Okay.
Now you have to imagine how each and every molecules can move themselves.
Yes. Okay.
That is made by this Ramachandran.
So how those molecules will move, he is predicting using this Ramachandran plot.
Yeah. So in our practicals also we are going to use it. So we have 3 more minutes. Can we have a quick practical now?
Okay.
So again, these are again technical terms. I'll make it fast. Actually, we need more time for a practical, but you go through all these basics are very, very important. Okay, Adi.
Okay. If we share the slides then
Definitely. And one more important, we got a recent Nobel Prize. We, in the sense, the people who are trying to use computers for understanding proteins.
AlphaFold, yeah, this is nice.
You know this?
Yes, I know that AlphaFold won Nobel Prize for in drug discovery itself.
So this is the field what we are doing now. Computational protein design. What we are doing, what you are committed for is a Nobel Prize oriented research. Okay.
Okay, yeah.
That to recent trend 2024. Okay, Adi.
Yeah, okay.
So you should feel appreciated, feel proud that you are learning something is in the current trend.
Okay.
And you go through this. This will be a third homework.
Review slides.
Go through your Nobel Prize work of
Okay.
Nobel Prize Chemistry 2024. What is the sign? What is their achievement in Nobel Prize Chemistry 2024?
Okay. I'll see that.
Please see. Okay. And again, these slides are so simple, like secondary structure, super secondary structures.
Okay.
Again, secondary will be further folded, like alpha, alpha, beta, alpha, beta, beta, beta. Clear?
Yes. Okay, sir. I'll see.
2 alpha, 1 beta, alpha is between 2 betas, or beta, beta, or 4 alphas, beta, alpha, beta. Correct?
Yes, I will check.
Yeah, please check. Yeah, and these are other important non-covalent interactions.
Okay, I will also look at this. Okay.
Yeah, please look at this. So last 1 minute, what we can see, you just I'll send some server name. Can you please open that and share your screen?
In chat. Okay.
Yeah, share your screen. Open some browser, please. In the browser, you click the link what I've shared now, just now. Yeah. Now you go to PDB. No, wait. We want some molecule to check. Okay. So I'll send a molecule in. Okay, how do I send? Go to PDB, protein data bank.
In Google. Okay.
Protein data bank. Yeah, you got it. Okay. You just type this word, what I'm typing. Or you type this. If it is not working, you type this 400 code. 5UA8 in PDB. Yeah. Now you click download files. Click PDB, legacy PDB.
GC or no?
Not GC, above one. Just click. Yeah.
Download.
And now upload that file. I'm not sure whether it will work because it's a small file.
Okay.
Run programs. You just click pro check. Can you see the written part? Later you can see, like, it will say the chemical property.
Okay, I'll check the later. Later I will see if it's written.
So you'll get a result now. Before you're running, no, you can see something is called pro check, and later you can see what it is written there.
Okay.
So you'll end up in a Ramachandran plot, Adhi.
Oh, okay. So essentially I'll get that.
Okay. If you are not getting that, you try with some other protein too.
Okay, I'll keep experimenting.
You just try to upload hemoglobin protein or whatever the protein you worked before. Okay, like that protein, you just try and check.
In my free time?
In your free time, you please try and check. Okay, and we'll try to learn more about it. I think it will take some like 1 or 2 minutes again. Fine.
Okay, sir.
And any doubts you have, you please post in the group.
WhatsApp when summer comes.
Fine, sure.
Okay, sir.
Okay, thank you, Adhi. Hope you have any quick doubts.
No, doubts? Sir. Today lesson.
Okay, hope you enjoyed, and you can find the notes in 15 minutes. Okay, I'll upload in the drive. Thank you. Take care. Bye.
Bye, sir.
Bye.